Dear students, please note down. These notes are available in the description of video. Please download your notes. In today's lecture, we will discuss plastids. And these plastids are double membrane bound cell organelles. These are characteristic features of all photosynthetic plants, algae. Note down, these plastids were discovered, and this term plastid that was coined by Ernst Haeckel in 1866. In these plastids, like mitochondria, DNA molecule is present. Due to presence of DNA, these cell organelles are semi-autonomous. As in case of mitochondria, the circular naked DNA is present that was double-stranded. Similarly, here in plastids, in chloroplast, the DNA molecule which is naked double-stranded. Generally, it is circular DNA, but it may be linear. Due to presence of this molecule, DNA molecule, these organelles are semi-autonomous. These have power of divisions. This term plastid that was given by Ernst Haeckel in 1866. These plastids are of three types. Leucoplast. Note down, leucoplast. These are colorless plastids, and these leucoplasts are involved in storage of food. These leucoplasts are further classified in three groups. Amyloplast, which store starch. Proteoplast or proteinoplast or alluroplast, which store protein molecules. And the third type is oleoplast, lipidoplast or eleoplast, which store fats. On the basis of storage of food, these leucoplasts, these colorless plastids, are of three types. Amyloplast, which store carbohydrates, starch. Proteoplast or alluroplast, they store proteins. Lipidoplast, oleoplast or eleoplast store fat molecules. Second type of plastids, chromoplast. Chroma means colored plastids, having color. These chromoplasts either derived from chloroplast or leucoplast. These are yellow or red in color due to presence of carotenoid pigments. And these chromoplasts, colored plastids, which are generally derived from chloroplast. At the time of ripening of fruits, the chloroplast, the green plastid, changes into chromoplast. It provides color to flowers for attracting pollinating agents like insects. At the time of ripening of fruits, the green fruits changes into a colored fruit, attractive fruit, to attract the animals for dispersal of seeds. And very important type of plastids, that is chloroplast. Dear students, in today's lecture, especially we will discuss chloroplast. These are green plastids. Note down, green plastid chloroplast. And these chloroplasts having three parts. One is envelope, second is this ground substance or matrix, and third component of chloroplast, these membranous structures which are named Thylakoids or lamellae. Chloroplast green plastid having three parts. This one is envelope. Chloroplast envelope is a double membranous coat. Both outer and inner coat या membrane are smooth, about 90 to 100 angstrom thick. Between these outer and inner membrane of plastid, chloroplast, a space is there, chamber is there, outer chamber is there, or this space is perichloroplast space or intermembrane space, which is 100 to 200 angstrom. Note down, each chloroplast having three components. One is this envelope, this coat. It is double membranous coat. Both outer and inner membranes are smooth, about 90 to 100 angstrom thick. And between these outer and inner membrane, a space, a chamber is there. It is perichloroplast space, or it is intermembrane space, which is about 100 to 200 angstrom. Second component of this chloroplast, that is matrix, or it is ground substance. Or not down NEET students, competitive exam students, this matrix is stroma, and the matrix or ground substance, which is mainly made up of proteins. About 50% part of this ground substance is protein, and the remaining part of the matrix that constitutes DNA, RNA, ribosomes, food particle starch, and plastoglobules. Note down, in this matrix the ribosomes, and these ribosomes are 70S type. Note down, these ribosomes in case of mitochondria, again 70S type, like bacteria. These ribosomes 70S type having two subunits. This smaller subunit it is 30S, and this larger subunit it is 50S. Both smaller and larger subunits collectively constitute a 70S ribosome. Here S is Swadberg unit. Later on I will discuss in ribosomes this DNA molecule. This DNA molecule, it is double stranded. It is naked without any envelope, and this naked double stranded DNA, generally not down competitive exam student in case of mitochondria as well as in plastid in case of chloroplasts, this DNA is generally circular. Occasionally it may be linear. In linear both ends are free, but in case of circular both ends are fused. And this molecule it is RNA. Ribosomes. Okay. First component, envelope, double membranous coat, outer inner, both membranes are smooth. Between these two membranes, a space is there. It is perichloroplast space, or it is intermembrane space, 100 to 200 angstrom wide. Second part is ground substance, matrix, or it is stroma. And the third main part of this chloroplast, these membranous structures, these elementary structures of chloroplast, and these are thylakoids or lamellae. Here at certain places these thylakoids are stacked over one above another. The stacking, the packing of these thylakoid membranes over one another, they constitute some units. These are grana or granum. Singular it is granum. This unit is grana, formed by the aggregation, by stacking of thylakoid membranes. And these grana, here this is three-dimensional structure of chloroplast. This one unit it is granum, or these are grana. In this grana, about 20 to 50 granule thylakoids are there. This 1 unit granum, it is formed by the stacking of thylakoid membrane, thylakoids, grana thylakoids or lamellae. In this 1 granum, about 20 to 50 granule thylakoids are there. And this thylakoid encloses a space or lacuna. It is inner space or it is loculus. Note down, these fine structures are thylakoids, and these thylakoids or lamellae, at certain places these are stacked, which are packed one over another to produce a unit that is grana. And in each grana about 20 to 50 granule thylakoids are there. And in this thylakoid a space lacuna is there. It is named loculus. And these grana are interconnected. And these lamellae which connects the grana are named stroma lamellae or intergranule thylakoids. Dear students, note down, competitive exam students, NEET students, these grana are absent in bundle sheath cell chloroplast in C4 plants. We have already discussed Karen's anatomy. Here note down, in case of algae, these grana are absent. So the chloroplast of algae or the chloroplast of bundle sheath cells, without these units, without these grana, such a chloroplast is agranal. But here in these photosynthetic plants, in all mesophyll cells, the chloroplast having grana, well developed grana are there. And these grana are interconnected by these membranous structures, these thylakoids. These are stroma lamellae or intergranule thylakoids. Their membrane is very important, very important. The membrane of this thylakoid, this is fret membrane, F-R-E-T, or thylakoid membrane. Note down, in this chloroplast, generally about 40 to 60 grana in one chloroplast. It means this entire structure of chloroplast having about how many grana? About 40 to 60 grana per chloroplast. And in each grana, how many thylakoids are stacked? About 20 to 50 per grana. And these grana are interconnected by these structures. These are stroma lamellae or intergranule thylakoids. These are some functions of chloroplast. Chloroplast, that is main center of photosynthesis, synthesis of food, in which consumption of CO2 and liberation of oxygen means this chloroplast. These plastids maintain a balance of CO2 and O2. In this chloroplast, synthesis of certain essential fatty acids. And note down, it is a storehouse of some food molecules like starch, lipids. Energy transduction. Chloroplast is center of energy transduction. Means one energy is changed into another form. This light energy, sun energy, that is trapped by this molecule, this organelle, and that sun energy is converted into chemical energy. In these thylakoids, certain pigment molecules are there, like chlorophyll A, chlorophyll B, xanthophylls, carotenoids. As well as in these thylakoids, coupling factors are there. to synthesize ATP. And these chloroplasts, at the time of ripening of fruits and in some flowers, these chloroplasts change into chromoplast to give rise color, to provide color to fruits, flowers, for pollination, to attract the insects, to attract the animals. This is all about plastids, double membrane bound cell organelles, having DNA molecule. Due to presence of DNA molecule having power of division, so these are semi-autonomous cell organelles like mitochondria. These organelles also show some similarities. Mitochondria, again double membranous structures. These plastids, chloroplasts, double membranous structures. Here DNA is there. In mitochondria, DNA is there. And here in plastids, the DNA is ctDNA, chloroplast DNA. But in case of mitochondria, the DNA, mDNA, mitochondrial DNA. But here in chloroplast, the DNA is ctDNA. Note down. And in case of mitochondria, as well as in this cell organelle, the ribosomes, and these ribosomes are 70S type in both mitochondria as well as in chloroplast. And here in this chloroplast, three components. One is envelope, second is this ground substance matrix or stroma, and the main important part, the elementary structure of the chloroplast are these thylakoids, lamellae, and the stacking of lamellae at certain places to produce grana. And a chloroplast without grana, like in algae, no grana. In bundle sheath cell chloroplast, no grana. That is agranal chloroplast. This is all about plastids. Three types of plastids: colorless, leucoplast; colored plastids, chromoplast; and the green plastids, chloroplast. And in case of red algae, in case of brown algae, these are named rhodoplast and pheoplast. Pheoplast in case of brown algae and rhodoplast in case of red algae. This is all about plastids, chloroplast, and in tomorrow's lecture we will discuss endoplasmic reticulum. Thank you.